Biomagic, let's learn quickly and very simply the Hershey and Chase experiment, class 12th. Hershey and Chase experiment, which is otherwise known as blender experiment. This was developed by Alfred Hershey and Martin Chase in 1952. And what they done, they are studying that DNA is genetic material or not. That is the main thing the experiment was dealing with, right? These two scientists worked with viruses infecting bacteria. That means bacteriophage. Phage is virus which is infecting bacteria, bacteriophage. So what they did, they grew some viruses on a medium that contained radioactive phosphorus and some others on medium that contained radioactive sulfur. That means they grew virus on two mediums, one having radioactive phosphorus, another medium with radioactive sulfur. Now, so the two mediums, the bacteriophage has been grown, that is a virus, radioactive DNA and radioactive protein. Now, radioactive DNA contained radioactive phosphorus P32, that is why the DNA became radioactive. And radioactive protein contained radioactive sulfur, that is why they become radioactive. Now, radioactive phages were allowed to attach and infect E. coli so that they can divide and scientists can find out whether the DNA is entering or the protein codes are entering. So, after infection, the bacterial cells were allowed to agitate in blender and then centrifuge. It is done to separate the viral coats and the other adhering phage components. That means to clear cut separating the different components. We can find out whether which radioactive element is shown in the blender after centrifugation in the pellet part, right? So these are the two mediums. In one medium, the bacteriophage contained the radioactive protein, that is S35. Another one where it contained the radioactive DNA, that is P32. In first medium, the bacteriophage allowed to infect the E. coli, and after infection, after the centrifugation and blending part, you can see there is no radioactive detected in the final cell. That is radioactive detected in the supernatant. That means in the pellet there is no radioactive. Here it shows that nothing is entered inside the cell which contained the radioactivity. Here the radioactive was shown in the protein coat. So this clearly showed that the radioactive protein didn't enter. Now in the second case where the DNA is having the radioactive phosphorus, after the blending and the centrifugation, it is seen that radioactive phosphorus detected inside the cells. No radioactivity detected in the supernatant. So in the pellet is the bottom part after centrifugation. Supernatant is the upper part. So in the pellet, radioactivity has been shown. So this shows that something which has radioactive entered inside the E. coli and divided, right? So the genetic material entered. So that genetic material is DNA. Bacteria which were infected with virus that had radioactive DNA were radioactive. That means where the scientists have put the DNA radioactivity, that means DNA contained the radioactive element, the bacteria were radioactive. And the bacteria that were infected with virus with radioactive proteins were not showing any radioactivity. So this clearly shows that the protein coat is not entering from virus to bacteria, it is the DNA which is entering. So DNA is the genetic material that passed from virus to bacteria, which is clearly seen in this experiment. So again I'll show the diagram. Here the protein which is radioactive element, and when after centrifugation you can see in the pellet there is no radioactivity, in the supernatant there is radioactive. That means the protein coat is showing radioactive. In the second medium where DNA was radioactive, in the pellet after centrifugation you will see that the radioactivity is seen in the pellet, which clearly shows that the genetic material which entered inside the cells is the DNA. So this experiment is very very important, which is showing that DNA is a genetic material. I already explained the bacterial transformation. If you want the video, you can see the video, you will understand. So these two are very very important in the class 12th syllabus. Hope you understood. Thank you.